right, well, it's 9 a.m. I'd like to thank everybody for joining our webinar. And yeah, we're very excited to discuss our new extraction-free coronavirus testing product. But before I begin, I would like to remind everyone that questions can be posted at any time by pressing the Q&A button at the top left of your screen. Simply click on that button and type your questions. At the end of the presentation, we will go through all the questions that are submitted and save plenty of time for discussions. So without further ado, let's get started. So this webinar will provide an overview of BioGX, provide an introduction of the extraction-free coronavirus test, which we are calling Xfree. We will discuss how Xfree will help laboratories increase testing capacity, review extraction-free marketing collaterals that will be provided to distributors, and as I said earlier, we'll provide time for question and answers at the end. So what I'd like to start off with is some company highlights for BioGX. Some of our attendees, this is the first time being exposed to BioGX. So BioGX is a pioneer in fully lyophilized assays. Currently, we have the largest menu of infectious disease targets, predominantly for the BD Max platform, but these are being ported onto other standalone real-time PCR devices. We are a fully integrated global business. We capture everything from R&D innovation, manufacturing, to product delivery. We have secured our supply chain currently for coronavirus products that will allow us to generate 2 to 4 million tests per week. For coronavirus testing, we have our sample to answer portfolio that includes our current United States EUA product that is available on the BD Max platform. We have recently released the coronavirus flu RSV combo for the BD Max platform. And today we'll be releasing the extraction-free coronavirus COVID-19 direct RT-PCR product. This will have both EUA and CE-IVD marking. Additionally, BioGX has, over the years, generated strong partnerships with companies including BD, Bosch, Thermo Fisher Scientific, Medline, as well as our 30-plus global partners. Now, to focus on BioGX proprietary sample-ready technology, all BioGX products are generated in this completely lyophilized testing format. So as you see from the animation here, all the components that are needed for real-time PCR are consolidated into one tube that's lyophilized. So the customer simply needs to do is just add water. And in this format, shipping is possible at ambient temperature. No freezing or thawing is necessary. The product, when it's built in these large lot sizes, has excellent reliability and reproducibility. And this format prevents the loss or waste of tests. Now here what we'd like to highlight is the lyophilized library that we've amassed over the past several years. So currently, what you see on this graph is BioGX being compared to 24 of the largest infectious disease diagnostic companies. And the legend here indicates the different syndromic tests that are available through these companies. Now, what I'd like to highlight is that currently BioGX has generated 185 targets for syndromic testing, with another 160 that are in the pipeline. In the upcoming year, BioGX will further increase our total library of lyophilized syndromic targets to 300 plus. Now, what I'd like to do is take a little step backwards and discuss the performance of one of our specific assays, lyophilized assays. That's the COVID assay for the BD Max that's currently available in the United States. Now, what you see here is a comparison of the BioGX assay, noted on the bottom here, and its limited detection. When this product was released, the limit of detection was published at 40 copies per milliliter. And you can see the other companies, Abbott, Roche, Hologic, Quidel, Cepheid, with their respective performance characteristics on their EUA-granted coronavirus product. Now, since then, the FDA has generated a reference panel where all vendors were allowed to partake in this to understand the performance with this specific reference panel. On this graph, what you can see is groupings of the limits of detection that were disclosed by each of the respective vendors. So what we see here at the bottom is a series of limits of detection that were grouped as low as 180 copies per milliliter, all the way up to 180,000 per milliliter. And what you see is the BioGX assay for the BD Max is amongst the top 4 groupings, where this standard panel reestablished the limited detection at 1,800 copies per milliliter. The metric that the FDA used was nucleic acid amplification test detectable units per milliliter. As a point of reference, the BD coronavirus product is currently at recognize the importance of offering template positive controls for every target within the assays we offer. And the format that's provided is what you can see here on the right-hand side. The lyophilized bead that contains the relevant RNA or DNA for the particular assay at 100,000 copies per milliliter. Now these controls allow for rapid validation as well as for use as in-run controls. Now, to briefly summarize, combining all of these aspects of BioGX, these are the hallmarks as to why customers choose BioGX and continue to work with us. Ease of use of our products in the just add water lyophilized format. The customer experience, where customers see the reproducibility, the robust performance characteristics. Clinical leadership. We have many laboratories around the world where the leading laboratories are consistently making use of BioGX products. The reliability and quality of our products, lot-to-lot consistency is one of the hallmarks of BioGX products. And the growing breadth of sample-ready products with our ever-expanding library. Now onto the focus of our webinar. So again, we are focusing this webinar on the newly released BioGX Xfree COVID-19 product that supports rapid extraction-free analysis for COVID-19 patient samples. This slide here highlights the simplicity of the Xfree COVID-19 direct RT-PCR product. The product comes in one lyophilized tube where you simply add water to rehydrate it. That lyophilized rehydrated master mix gets aliquoted into a multi-well plate. Patient sample is mixed, seal is applied, and that plate is then added to the real-time PCR device. and run. This is approximately 80 minutes for the real-time PCR instrument to complete its programmed run. Now, with this direct sample addition for extraction-free processing of COVID-19 patient samples, laboratories are now able to further unleash their testing capacity. Now, again, this product allows for direct patient sample addition, or in cases where laboratories would like to use extracted nucleic acid, we have validated both sample types. And they are available for use on the Bio-Rad CFX 96 platform, as well as the ABI QuantStudio 5. Additionally, customers are open to validating on comparable platforms that contain the same optical sensitivities, optical capacities. So as I touched on earlier, the Xfree COVID-19 product, which will have EUA and CE-IVD marking, will be released today. What we have in our pipeline is for more extraction-free products to be generated. These will include the COVID flu product, planned for release in Q1 of 2021. And a little bit further in our pipeline, we plan to apply this extraction-free technology to other respiratory panels, as well as to UTI panels. Again, this format allows for sample to answer capabilities on a variety of commonly found real-time PCR instruments within laboratories. Again, in times of constraints where extraction reagents are in short supply, this product allows you the flexibility to use either direct patient sample, or when extraction kits are available, to make use of extracted eluate for template. So the clinical concordance study that was performed with the extraction-free product made use of 77 patient samples, 41 of which were identified as positives, 36 were negative. And the comparator tests that were used for this clinical concordance study was the current FDA EUA product that's used in the United States. This is the BioGX SARS-CoV-2 assay for the BD-MAX system. Additionally, for extracted patient sample, we made use of the CDC EUA method that recommends the QIAGEN QIAamp DSP viral RNA extraction kit. And this was used with the BioGX Xfree COVID-19 product. As you can see, for all occurrences except for the BioGX, the BD-MAX comparison, with one of the 41 patient samples that was missed, you could see 100% agreement. Again, going back to this 98% agreement, what we saw here is that it was a positive patient sample that was detected at the limit of detection on the BD-MAX assay. So we have great confidence in the performance of this assay when patient samples are obviously within the limited detection range, which I will touch on in the next slides. To further discuss the robust performance of the Xfree COVID-19 assay, I'd like to highlight here the performance of the test as visualized on the BD-MAX assay that's used in the United States. Here you can see all the positive patient samples that were run on the BD-MAX platform. And those same patient samples that were run as direct input with the Xfree product on the QuantStudio 5. And those same products, again, as direct patient sample input on the Biorad CFX96 Touch. Again, you see robust amplification, and it makes use of, as I touched on earlier, real-time PCR platforms, specifically those that have 5 optical channels. Within this assay, we make use of FAM, Texas Red, and Quasar 705. The limited detection for direct patient samples in the Xfree product is 2,000 to 4,000 copies, and this varies based on the instrument and the sample type. What we've seen for eSwab, in cases we have a 4,000 copy per milliliter LOD, and in certain cases we see with saline samples, the limited detection will go up to 4,000 copies per milliliter. When using extracted patient sample, the limited detection for the Xfree product is 1,000 copies per milliliter. To further highlight the leading sensitivity and reliability of the Xfree product, here we can see the comparison to the currently available Quidel product that allows for direct patient sample input. In this case, only a dry swab is the accepted sample type. And with this sample type, Quidel has published a limited detection of 12,800 copies per milliliter. Conversely, the BioGX Xfree product has a limited detection of 2,000 copies per milliliter for many of the sample types. And as I touched on earlier, in certain cases, it goes up to 4,000 copies per milliliter. And one of the hallmarks of the Xfree product is the fact that it can make use of all major collection devices. This includes UTM, VTM, saline, as well as eSwab collection. Now, the Xfree product helping laboratories. The format that we have generated the Xfree reagents in allows for fast and simple workflows and is designed to help laboratories increase their testing capacity without additional expenditures to hire expert personnel or to invest in new automation. Now, how is this done? The easy-to-use format, again, as I said, a single vial containing all components for real-time PCR is simply rehydrated and dispensed into a multi-well plate. It's fast and effective. No sample preparation is needed, along with the broad coverage of sample types. You're not limited to certain sample types. And the high volume, high throughput workflow. So for typical 96-well, 5-channel real-time PCR platforms, the 80-minute turnaround time for the run allows for 2,000 samples per 24 hours to be processed with a 96-well block, or up to 6,000 tests per 24 hours if the laboratory is using a 384-well block platform. Additional benefits are cost savings, increased capacity without adding expert staff or new automation, accessibility, the compatibility with direct and extracted patient samples, and the multi-platform aspect. Again, we've validated this on the QuantStudio 5 and the Biorad CFX96 Touch, with plans to add more validated platforms in the future. In the next few slides, I'd like to touch on the marketing collaterals that will be provided to the distributors. So here, our first marketing collateral is a one-page flyer. And this is a quite dense flyer that summarizes all the key aspects of the Xfree COVID-19 direct RT-PCR product, highlighting the extraction-free approach, the simple 1, 2, 3 steps that are needed to set it up. Simply rehydrate, add patient sample, and add the sealed plate to the real-time PCR instrument. The highlights of the test are summarized as well. The fact that no other reagents are required, direct sample input is accepted, and the high throughput workflow that is capable with 96 well or 384 well blocks. Again, the dual marking, emergency use authorization only, as well as CE-IVD, and the summary table for the clinical concordance that was generated. Additionally, BioGX has generated a dedicated web page for the extraction-free product. And this is a screenshot of that web page. Once you are on that web page, you also have access to a quick video that highlights the simplicity of the workflow. And you also have a quick guide that can be downloaded, instructions for use manual, all the run files that are needed for either the Quant Studio or Bio-Rad CFX, and fact sheets. These are healthcare provider as well as patient fact sheets. Additionally, we have a marketing collateral that is one page that is a quick guide for quick reference for laboratory staff to have an overview of the workflow to begin testing. Again, this workflow is compatible with 96 well and 384 well blocks. The first panel you can see here highlights the direct sample workflow. Again, we are highlighting the sample types that have been validated and the simplicity of the workflow. Rehydration of the master mix, dispense into multi-well plate, addition to the real-time PCR platform. Additionally, should the laboratory choose to run extracted patient sample, again, we see all of the sample types that have been validated and the same 1, 2, 3 workflow. What I'd like to highlight in this slide is the added value that is built into the Xfree product, where, as you can see here, a 400 microliter rehydration requires 24 dispenses of 15 microliters into the multi-well plate and 5 microliter addition of patient sample. With purified nucleic acid, as you can see here in the extracted sample workflow, 400 microliters is used again to rehydrate the master mix. But now, since the patient sample has been purified of any sort of inhibitory components, less master mix can be used with 5 microliters of eluate. So the added value is that
36
patient samples can be processed should there be the need to use extracted nucleic acid. And lastly, we have a 2-page brochure that provides a bit more detail, highlights the properties of the Xfree, the benefits of using it, the sample types, as well as a simplified workflow and the clinical performance. And that was a quick overview of BioGX as well as the Xfree new product for coronavirus testing. And with that, I again thank you for your time and your interest in the new Xfree product. Look forward to further discussions with each of you in the future. Thank you very much.